Hello and welcome to Understanding Emotions. My name is Sankalp. I recently completed my PhD in psychology/neuroscience from the University of Oxford. And before I begin, I must stress that this is a personal channel. So, whatever I'm going to talk about here is, and whatever I do generally on the internet, is separate from my research role at the university. All right, so what we are going to do today is talk about human memory, actually, memory in general. And the reason why I say that is because a lot of neuroscience, when we talk about hardcore mechanisms, is actually done in animals. So there will be some inference involved from animals to humans, but we'll cross that bridge when we get there. To be clear, there is studies done in humans as well. So there's good reason to believe everything that I'm going to talk about today will apply to humans. But before that, just the format of this live stream, just so that you know. So the format that we have for today's live stream is what I'm actually doing is in fact recording a video on memory, which will be like this long video, meaning what's going to happen is I will press the record button, I will record and I'll sort of redo takes if needed. And the intention is to ultimately make like a nice edited video on memory, which includes everything we talked about last time. And then today's will be the mechanism. And then the third video will be application. So how do we use this? Excuse me. If you're live, you will have to deal with my burps because I normally eat a little bit before this one, and it's a bit of an awkward position to sit in. Honestly, the chair is not ideal, blah blah blah, but we'll handle this in the larger service of science. I also have this thingy here, which is clearly not very well positioned at the moment. Okay, something like that should work kinda. Yeah, I will probably also run out of paper today, so I don't know how to deal with that necessarily. But again, we'll cross that bridge when we get there. Okay. Cool. So if you don't know where we are, in the last video, in the last live stream, I talked about what memory is and indeed the different types of memories. And in this episode, I'm going to talk about the mechanisms. Before we even begin, it's sort of important to get a sense of get us motivated about why memory is so important. Like, look, we are all human beings, and our sense of self, a sense of being human, a sense of living is heavily predicated on our memories. Like who we have been in the past, what kind of people we are, like who are the kinds of people we have spent time with, and what are the places we have visited. So who we are is heavily predicated on memory. If you have someone who is amnesic, in other words, if they have lost their memories or if they can't form new memories, then you begin to think that something about their selfhood, something about their personhood is lacking. And obviously you get these heartbreaking moments where someone has dementia and they forget who their child was, or they forget who their close cousin was. And that sort of a thing can be quite heartbreaking. So memory is not just, who can remember like 100 digits of pi or something. Of course, that is the kind of memory that is also important, like just retaining information. But memory is also just who we are and what our sense of self is in this world. So we're gonna talk about all of that. And hopefully this motivates you into thinking that, look, indeed, memory is super important. And it's indeed something we should be spending time in understanding. And of course, there's this whole component of some memories we don't want to have. Like, for example, if there's something traumatic that happened in childhood, or something traumatic that happened which may not be like a clinical level trauma, but something that bothers us, and we'd rather not remember it, or at least not have the emotional charge associated with it. And so we are going to talk about that as well. And finally, there is this element of performance. Like, if you want to learn something, if you want to perform, then chances are that we need to hold certain amount of information in our brain in order to be able to make connections. And any productivity, assumes at least some level of memory. Like if you can't hold any bits of information in your brain, then what are the chances that you're going to synthesize them and then, you know, like merge and mix and match them and create something that is truly valuable? Chances probably are none. So human memory is also important in terms of performance and learning. And we're going to talk about that. And finally, there's this aspect of, you know, memory changes a lot with technology. Like, you know, back in the olden times, people had to remember phone numbers and whatnot. And now we sort of offload it, right? Like we give our phones that. And until like perhaps the last year, we needed to at least remember information in terms of if you're a knowledge worker, or if you want to, you know, like if you want to write an email, you would have to remember what you want to say, and you want to remember the past history and so on. But right now with AI and everything, you have the world's information at your fingertips. So memory is also important for that kind of thing. So hopefully that gives you a sense of why memory is important. And what we're going to do today is focus on the neural mechanism of memory. What does neuroscience have to say about memory? And it's some of the most fascinating, some of the most mind-blowing, some of the most mind-blowing research that has ever been done. So today's video, at least from me as a neuroscientist, is so beautiful, it's so powerful, it's so interesting. And I hope to share that enthusiasm with you. And let's get started. So memory today. Okay, let's go. All right. So once again, if you are live, I just want to emphasize that what I'm trying to do is record a video, which means sometimes I'll say things again and again. Sometimes I will pause in between, do whatever is needed to serve that video. But if you are live and if you have questions, you can put it down in the chat. And whenever I take a pause or something, I might have a look at it. Oops, that was not intended. And that kind of thing. So hopefully, let me just actually type it in the comments. If you are live, know that I'm recording a video on memory, on the neuroscience of memory. But if you have questions, I will glance at them, reply to them in breaks or in between recording sessions. So yeah, do post your comments. That was me dictating, by the way. Oops, it looks like I have exceeded. But if you have questions, I'm going to reply to in breaks or between recording sessions. Okay, that sounds good. Boom. Posted. Fantastic. Okay. Sounds like I'm recording as well. That's wonderful. Hopefully you can see this part of the screen fine. Great. Okay, dokes. Awesome. It sounds like I was I should have ideally been talking with the full camera. But anyway, we're going to talk about the neuroscience of memory, the mechanisms that are involved in memory. So if you were with me for the last section, we talked about this one patient, HM, who had like major parts from the middle of his brain removed. And the reason that the surgeon decided to take that decision is because HM had seizures. So he had these epileptic seizures that were just outside the normal range, and this was uncontrollable and just terribly affected the quality of life for HM. And so therefore that part of the brain was removed. Except once that was removed, they found that HM lost the ability to form new memories and just became a little bit of a neuroscience test subject. And what HM showed us is the critical role of the hippocampus, which is a structure in the middle of the brain, which was removed in his surgery, in human memory. It also showed us that, you know, memories come in different shapes and forms. There's no such thing as one kind of memory. But the idea that memory is sort of focused, there's like a librarian in your brain which takes in new memories and consolidates them and then files it in different parts. That is done largely in one section of the brain called the hippocampus. And today we'll see there's an adjacent region which was also removed called the amygdala, and another region called the entorhinal cortex. We're going to talk about all of that today. And really, we're going to zoom in on what are these regions of the brain really doing. Like what are they computing? What do I mean by this is a librarian? How do we improve our performance? How do we improve our learning and memory? We're going to all cover that quite in detail today. So I'm quite excited for this one. Okay. So let's get started. So in order to understand human memory and how the hippocampus supports memory, I shouldn't say human actually. Let's do this again. So in order to understand memory and how the hippocampus supports memory, we will now look into the mechanism of what the hippocampus is actually doing. And the understanding of what's happening inside the hippocampus actually led to a couple of Nobel Prizes being given out. So this is like super fascinating, important research. So in order to understand the mechanism of how memory works, we need to understand this type of the cell that is involved in the hippocampus and understand what it is doing. So John O'Keefe is the first person who discovered this. And what we find is that inside the hippocampus, there is something called as place cells. So I'm gonna just write it down here. Hopefully you can still see it. Boom. We're gonna talk about place cells. Place cells. And this is like a type of cell that is involved in the human hippocampus, and indeed in hippocampi, be it animal hippocampus, human hippocampus, and whatnot. And we'll see that a lot of studies are actually done in rats and mice and rodents, and then we will also see some analogs in humans. So first of all, what are these place cells? So we need to talk about this one experiment done by John O'Keefe, and I'm going to pull out my notes so that I have the exact dates also in front of me, not like literal dates when it was performed, but just to give the right sort of citation. So place cells were discovered in 1971, and this was by John O'Keefe. And the idea was, I'm not going to describe the exact experiment. I'm going to try and describe conceptually what happened. So the idea was, what they found is that, let's say if you have a rat and the rat is allowed to navigate through this one-dimensional sort of tube. So imagine like this is like a track, a race track of sorts. And what you're doing is looking at this track from the top. So this is like the runway. Imagine this being like a runway, like an aircraft runway, and you're looking at it from the top. And let's say there is like a rat here, which is moving along this track. So for example, if I draw my rat here, boom, 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 boom. Yeah, that's the rat. And it's moving along this track. So what they found was that if you look at the hippocampus, so if you take brain recordings from the hippocampus of this rat, what they found was that there were certain cells that fire at certain places. So for example, if you look at one of the cells inside this rat's hippocampus, it would only fire at sort of this sort of location. And technically the place fields are like circular. So if you imagine a two-dimensional grid where the rats are moving freely, then you will notice that there is one cell in the hippocampus would fire at some particular location. So for example, this sort of a circular region, and there are the cells that fire at other locations. So what this tells us, this is like quite an important discovery, because what it tells us that there are cells in the brain that respond to specific locations. And these cells were called the place cells. They received a Nobel Prize for this discovery. And why is this such a big deal? I mean, first of all, we take it for granted now that we know, but to have like a dedicated module in the brain that's keeping track of where you are in space, this is super important, because this tells us that the brain might potentially be maintaining a map. Remember, these cells fire independent of the direction in which the animal is coming from. So if the animal So for example, if the animal comes from the top to here versus the bottom to here, whenever the animal is in this location, there would be certain cells that would fire. And those cells are what, therefore they were named as place cells, because they seem to fire in specific locations. And the second thing, therefore, we need to now know is also what's called as grid cells. And again, I told you that these place cells are independent of where the animal is coming from. They just fire all the time in this position, meaning there's some sort of a map that the brain might be maintaining. So there would be some cells would fire in this location, some cells would fire in this location, some cells would fire in this location, and together, taken all cells together, they would form like a bit of a map of the environment. And then later there was something called as grid cells that were discovered, and this was by the Mosers, Edward and Maybrit Moser, and these people also, actually they all got the Nobel Prize collectively for discovering these. They discovered something called as the grid cells. So if place cells tell you where you are in a certain point in space, grid cells tell you a little bit of the coordinate system. So they are like mapping a coordinate system. It's also important to know that while the place cells were found in the hippocampus, grid cells were found in an adjacent area, which was also removed in HM surgery, called the entorhinal cortex. And so what is the difference between place cells and grid cells? So while place cells fire at specific locations, grid cells fire in multiple locations and they form like a grid-like pattern. So for example, if one cell is firing here, maybe I should make it a bit bigger. If one cell is firing here, the other cell would fire perhaps here, perhaps here, perhaps here, perhaps here, perhaps here, then here again. And I'm not doing necessarily the best job in drawing. And if you're on audio, I just drew like a couple of circles, which form like a grid-like pattern. So if you imagine again like a two-dimensional surface, and if you're looking like a football field, and you have like a drone flying over the football field, and if you look down, you would notice that place cell would fire at one particular location in that football field, maybe at the goalpost, whereas grid cells would fire in a grid-like pattern, so in multiple locations throughout the football field. And this would be creating some sort of like a coordinate system in the brain. Now, interestingly, when the grid cells did fire, it was not like a Cartesian coordinate system in which, like we were drawing, like they were like not firing in neat X and Y axis, but instead they were firing in some sort of a hexagonal pattern, meaning there was like 6 vertices to the firing pattern. So if you can see it, if you are watching the video, you can see I've drawn like a bit of a hexagon here. And so the idea here is this thing, if you look at it, it forms a bit of a hexagon. And this is what grid cells showed, this beautiful firing pattern, which looked like a hexagon. So hopefully these two types of cells tell you that the brain is maintaining some sort of a map of the environment. And not only is it maintaining a map, it's maintaining this map independent of the animal's location itself. So the technical word is allocentric, which is opposed to egocentric. So egocentric means relative to me, whereas allocentric means just absolute in terms of the space itself, relative to like a neutral observer, let's say. So the brain is maintaining some sort of an allocentric map. Now, why is all of this important for memory? Like, I think when we want to understand memory, we need to understand one of the most common use cases of memory is in fact navigation. Like going to the supermarket down the road, you have to navigate to that shop. And somewhat the most basic form of memory is indeed through navigation and space. So if we could understand how we keep track of space, then that's a good first hint to think about how we keep track of emotional memories. And we are going to slowly build up to that. But the first thing to understand is indeed place cells and grid cells. Place cells fire at certain locations. Grid cells fire in a grid-like hexagonal pattern. And together, these two provide the coordinate system and specific location for an animal to navigate their environments. And once again, this discovery of place and grid cells in the hippocampus and entorhinal cortex respectively won the Nobel Prize. And John O'Keefe and the Mosers won the Nobel Prize for that. So great job to them. Clearly, they have informed a lot of our understanding of human memory going forward. Okay, phenomenal. Okay, so those were place cells and grid cells. Hopefully that explanation was good enough. I'm just going to check that I have not missed something in this section before moving on to the next section, which is even more fascinating. Yeah, fascinating. Cool, cool, cool. Fantastic. Okay, cool. So now, ladies and gents, it's going to get even more interesting. Let me ensure that I am indeed recording. Okay, let's make this even more interesting. Actually, let's stop this for a minute. All right, so we're going to now talk about something called as replay, which is, how memories get consolidated. I'm just going to refresh my knowledge about it. There's a lot of material we're going to cover today. So get set and go. Okay. Play during sleep. Okay, fantastic. Okay, let's record. Okay, so now that you understand place cells and grid cells, we are going to move on to an absolutely fascinating, mind-blowing discovery that were made in the hippocampus, specifically related to place cells. So let's tell you what this experiment is and tell you why this is so mind-blowing. So on the screen, you're seeing place cells and grid cells. And what I'm going to talk about now is what's called as replay. Replay. So what's happening is, imagine again, you have this one-dimensional track, and I say it's one-dimensional because, it's like the animals could only move on like this sort of horizontal axis. And again, you have a nice rat moving, and I can see the quality of my rat is, the rat drawing is going down and down. And so if you're listening on audio, what I've done is, again, imagine like an aircraft runway, you have a drone floating above the runway. So you're looking at like the flight moving, and it can really move in this one direction, right? And the same goes with a rat. So I've drawn on the screen a rat, and you're looking at it from the top down angle. And we already know that there would be certain cells that would fire at certain locations. So for example, there would be one type of a cell that would not fire anywhere here, but the moment the rat reaches a certain location, it would increase its firing. So it would like go up like that. Right? So what's happening is that, let's say in this location, a set of cells A fire. Let's say in this location, a set of cells B fire. And let's say in this location, a set of cells C fire, right? So this is again recording from the rat hippocampus. I'm pretty sure the same is true for humans. But let's say there are these A, B, and C cells that are firing in this particular location. There is a very fascinating discovery that I believe Wilson made. I'm going to ensure that I get the citation correct. Okay, so there's a very fascinating discovery that was made in 1994 by the scientist called Wilson. And they found that when you take this rat and you train this rat to go through this maze, and you notice that ABC cells are firing, when this rat is later sleeping, so let's indicate it by ZZZ. When this rat is later sleeping, let's draw the rat again. What you find is that the same set of cells, A, B, and C, are also firing. So this is again, to me, it's mind-blowing. So when this rat sleeps later at night, quote unquote, there would be like these same sets of cells that are firing. And not only do they fire in like a very quick time window. So while the animal was doing the task in real time, when this animal was navigated this airport runway, so to speak, the set of cells A, B, and C fired maybe on the order of seconds. So, this might be something like 10 seconds. When you look at the activity from the same region of the brain in sleep, this thing would fire in 100 milliseconds. So that's like really, really compressed and orders of magnitude less. So the same set of cells would fire in 100 milliseconds or so, that sort of a time range. So what this is suggesting is that when we are sleeping at night, the brain is taking what we have done during the daytime, the challenges we have navigated, the new paths that we have learned, in this case navigation paths, but you can imagine this for any other path that you might have navigated. Perhaps it's a new subject that you are learning, and you're learning the various steps that goes into creating this proof. If you're into mathematics, or if you're into banking, all the steps that are needed to file your accounts, I don't know, whatever this thing that we are doing in the daytime, in the evening, what's happening when at sleep. And again, in animals, they may or may not maintain like the circadian rhythm that we humans maintain. But regardless, whenever the sleep is happening, what's happening is that the same set of cells are firing in a very rapid compression. And this might be the case why, dreams are so like rapid and rigid. It's completely speculative. But what we know for sure is that the same set of cells are firing when the animal is sleeping, suggesting some sort of a consolidation is happening, suggesting that we are like taking all of these memories and putting them all together and learning from them and making these connections stronger. Now, so that's the first thing. Later in 1999, Nasady is the name of the scientist. And there's always a team, by the way. I normally just take name of the first author. In fact, it's the last name of the first author. But in 1999, in the Journal of Neuroscience, they published this fascinating finding, which is not only that at night the same set of cells are firing, but in fact, they are firing in the same sequence. Meaning, if the cell A, B, and C fired during the actual task itself, the sequence A, B, and C fired in that sequence at night in rapid compression. So not only did the cells fire, but the same sequence was maintained. Now again, to us, this might seem obvious and like what other way it will fire, right? But that's a discovery that needs to be made. Perhaps, you never know, in an alternate reality, they might fire in like random points. But indeed, the sequence is maintained when it's at sleep. So again, to me, this is fascinating. Okay, so now that we know that the same set of cells are firing and they fire in the same sequence, the thing is, it doesn't end there. Okay, so now that you know that the same set of sequences, let's do this again. Okay, so now that you know that the same set of neurons are firing in the exact sequence, be it at rest, or the exact same sequence when it is done in the task. I think I need to be more clear on that. Let's do this again. Okay, so now that you know that neurons maintain the same sequence at sleep that they did during the task itself, we know that there is some sort of a replay happening in the brain. This phenomenon is often called as the hippocampal replay. But the thing is, it doesn't stop there. It absolutely doesn't stop there. And what the researchers found, and I want to make sure again that I get the citation correct. So what the researchers found in 2006. So remember, this discovery was 1994. And then the sequence discovery was 1996, I believe, or 1999. And then finally in 2006, what they found was that when the animal, let's say if it reaches the end of the goal and they give them a reward here, perhaps a fruit juice. And again, for those listening on audio only, I'm drawing an animal at the end of the track now. So imagine an aircraft at the end of the runway. And at that point, the animal was given some sort of a reward. Let's say they were given like a fruit juice. And then at the end of the runway, when the animal was sort of like resting, sort of did the task, achieved the goal, what they found was that there was a replay happening even in those moments. So not only is there like this consolidation replay step happening nicely in sleep, but even when the animal is at rest while doing the task. So for example, imagine I'm learning something, but I take a little bit of a break. what happens is my hippocampus is going to revise that sequence that I'm doing. And same for the animal. If you record from the electrodes, they found that the same ABC style of replay is happening towards at moments where the animal is at rest while it is awake. And again, the same sort of compression sort of thing holds. So this is quite fascinating. And what is also observed is that this replay is not like ABC, but this replay is actually CBA. And this is often known as like a reverse replay. Reverse replay. Just writing reverse replay on the screen here. So at this moment, what's happening is not just ABC, but also CBA. Now, look, I'm being a little bit, I'm generalizing a lot. Like there's many moments in which this can happen. So for example, replay can happen, not only at rest, but during the track and so on. But we are also tracking the trajectory of the discovery. And at moments of awake rest, especially towards the end of the goal, there is like a reverse replay that's happening. And so the question would be like, why would the brain replay the sequence in reverse? What is that serving us? And one hypothesis could just be it helps remember better. If you go from forward to backward and backward to forwards, maybe that sort of a ability to traverse the path in different ways, that just helps remember the path better. But the other interesting explanation that's given out for this is because more often than not, reverse replays is observed at the time of reward. So after the animal has received some reward. One explanation for reverse replay is what's called credit assignment, meaning the animal, once it gets the reward, needs to know what is the path that produced that reward so that I'm able to repeat that path in the future. And this sort of a reverse replay helps me sort of chart my course back. So if I received some good things happened to me at this point, I need to like just go back in time and rewind back and see what is the things that I did to cause that good thing to happen to me. And so that's the idea behind reverse replay that generally happens at awake immobility, awake rest, during times of rest. And this gives us some idea that, hey, look, maybe when we are learning new things, it might be useful to think about rest periods as strategically enhancing our memories or enhancing our performance. And this gives us some hints along that. Now, finally, something to also mention is that, of course, reverse replay happens, but another fascinating finding, and this is 2007 Nature Neuroscience, the one of the authors is Diba, and what they found is that once the animal has learned this trajectory, we know that at the end there is a reverse replay happening. But at the beginning, even before the animal starts walking on this trajectory. So again, remember, if you're looking at a runway from the top down, this is the start. And when the animal has sort of learned this trajectory, it has done this a few times. What they find is that even at the start, even before animal takes the first step, it starts replaying the sequence again. And this seems like a little bit of an analog of planning. So imagine the animal as the start point, it sees the reward at the end, and in its brain, it's already replaying those sequence. And when I say replaying, the exact set of neurons that are activated, let's say in ABC, are now activated in the animal's brain. So if I draw a dialog box here, there will be like an ABC sequence that's happening even at the start. And this is considered like perhaps some sort of a neural correlate of planning. So now that I'm at the start of the sequence, I can see at the end, and I've done this a few times, I can actually begin to rehearse that sequence even before I start. And that's some sort of a neural correlate of planning and like sort of this sort of long-term, well, in this case, 10 seconds is the term, but perhaps we can extend that and think about this could be the basis in which how we make these longer term plans. So hopefully that was interesting. Again, to me, this absolutely blows my mind. All right. Fantastic. Cool. So this section looks good. If you have any questions, please do ask. This is the kind of moments in which I will take some questions and that kind of thing. Cool. Fascinating stuff. Okay, I think we have done a decent job to cover replay. It's not easy at all. Okay, I think we are ready. Okay, now to summarize. So far we have seen that there are place cells and grid cells in the brain. Place cells fire at certain locations. Grid cells fire in a bit of a coordinate system, so they fire like a grid. And together these provide location as well as the coordinate system to the brain. Now, when we look at these place cells and when they fire at specific locations, you can think of them as the animal building like a trajectory of what gets it rewards, what gets it success. And the more fascinating finding is when this animal is at sleep, when this animal is sleeping, the same set of neurons in the same sequence are firing at night, suggesting that sleep is not this passive process that we just go to bed, but there is like memory consolidation happening. There's learning happening. You're learning about the world. And then later we also find that not only does this sort of a consolidation process happen at sleep, but it happens even in rest periods when the animal is resting during it is doing the task itself. So for example, if I'm learning something, if I take a little bit of a break, chances are that my hippocampus is going to repeat and replay the information that I have been learning. So now what we're going to do is talk about something called as phase precession. And do not worry, I will explain it as well as I could in as accessible a language I can. So far we have been focused on these rest periods, and we have shown that these rest periods are not passive periods, but instead the animal is learning, the animal is actively consolidating, and so on. But what happens during active exploration itself? What happens when the animal is actively exploring? Is there any sort of consolidation on memory process happening during that period? Like how is this memory built? So we're going to come back to our diagram here and talk about, again, let's say the animal is navigating this trajectory. Oopsie doops. So let's say the animal is navigating this trajectory, and instead of now drawing like a full runway, I'm just going to draw a line because you get the idea, I suppose. What's happening is, first of all, there are waves of activity happening in the brain. So imagine like the activity going up and activity going down, activity going up and activity going down and whatnot. And this is not necessarily specific to place cells. This is just like general activity. The brain is not like flat. It's like it's cycling. And so these are like brain neurons firing. So what happens now is that when the animal, let's say, is in this position, let's say the place cell associated with this position is firing here. And it could be to the left or right. It doesn't necessarily have to be at the top. But what happens, let's say this is like A. But what happens is when this cycle sort of repeats, and let's say if the animal is in this position, and this is B, let's say, what's happening is that the A would happen slightly before and the B would happen here again. And then when the animal is in, let's say, position C, there is something like this that is observed. And remember, this is now during active exploration, and these are neurons firing. So when the animal is in position A, the set of neurons corresponding to the place field A fires. But when the animal goes to the next position, it sort of makes room for the set of neurons corresponding for B firing, but also the previous neuron also fired. And so what's happening is it's not just during sleep that this sort of a sequence is building, but it's happening during active exploration as well. And perhaps this provides the building blocks for the replay to happen either during rest or during sleep. So even during active exploration, the animal is constantly building this map of the environment. Hopefully that made sense. Boom, boom, boom. So what this tells us perhaps is that learning and this process of consolidation happens. So what this tells us perhaps is that this process of encoding a sequence, this process of memorizing a sequence starts not only in sleep, but it happens during the task itself. And perhaps there are like these two modes of the brain. One mode is where it's encoding, when it's building information, and when it's sort of the hippocampus is receiving information and building the sequence, which is the active exploration phase. And during the periods of rest and sleep, the brain compresses these sequences and begins consolidating it, and then sends it to long-term storage, which happens in other parts of the brain. So this is the mechanism by which it seems like the hippocampus and the entorhinal cortices are working. So this is the mechanism by which the human hippocampus and hippocampi in general seem to work. Okie dokes. So that was phase precession. Hopefully that was all right. Hopefully that was all right. So again, we are by no means done. So far we are still in, we're not even halfway through, I think. Well, maybe we're halfway through. Cool. So you talk phase precession. The next thing we're going to talk about is something called as sharp wave ripples. But I might need to pee before that. So let's see. All right, folks. I need to pee. So what I'm going to do is come back in 10 seconds. And we're going to talk about something called as sharp wave ripples. So there's some like causal evidence that we found later on in science, which is quite important. So we're going to talk about that. If you have any questions, let me know. There is sharp wave ripples, reconsolidation. Again, another fascinating study. We're going to talk about abstract spaces, emotions, and the amygdala. And that will be today. Cool. Lots of interesting things coming ahead. Let me just free my legs a little bit and free my soul from the need to use the restroom. And I will be back in a second. Okay, hello, what's up? So hopefully now you have begun to appreciate the fact that the hippocampus is quite a smart region. Okay, docs. Cool. Hopefully you can hear me all right. So hopefully by now you are getting a sense that the hippocampus is quite a smart region. Am I audible enough? Okay, so hopefully by now you understand the hippocampus is quite a smart region and it is helping us shape and form new memories. We are now going to talk about this process of consolidation. We're going to talk about this process in details and how is it that something like sleep improves our performance and the causal evidence for that. So one of the things that we have discovered in the recent past, and again, I want to ensure that I get the citation correct. So the first thing to understand about this, so what is consolidation in the first place? So these scientists, Franklin and Bontempi in 2005, they proposed that hippocampus serves like a bit of a librarian, a bit of a temporary index. And what it's doing is once it gets these memories, it takes this process of consolidation is basically the process of taking these memories and then storing them in the long-term storage, which is normally our cortex. So these are like, parts of the brain that are towards the outside. So what is this process of consolidation? One proposal given by these scientists, Franklin and Bontempi in 2005, is that the hippocampus acts as a bit of a librarian and the cortex acts as a bit of a storage. So think about it this way. When new memories come about, when new memories happen, the hippocampus is taking these memories, replaying them forward, backward in the way that we described in the previous section. And then at rest, it's taking those memories and putting them into long-term storage. And this long-term storage is normally in the cortex. If you don't know what the cortex is, this is like the general catch-all term for more of the outer part of the brain, which we consider is more evolutionary recent. So humans have more of these outer folds compared to other mammals. And it seems like the hippocampus is taking these memories and putting them into cortex. And this process is what's called as consolidation. But so far it is just theory. And a critical proof for this theory came in 2009. And this was the scientist named Girardeau, 2009, pretty much butchering the name, but hopefully it's still all right. And this is what they did. So first of all, there's something called as sharp wave ripples that we need to understand. So I'm going to ensure I'm on screen. So there's something called as sharp wave ripples that we need to understand. So the hippocampus, remember, this is the region that was removed from HM's brain. It's not like one thing. There are like many parts of the hippocampus. And the two important parts we're going to talk about are what are called CA3 and CA1. So what's happening is that the CA3 starts firing normally first and then CA1 fires. And so what happens is if you record from a lot of neurons from CA3, you will notice that, there's like a wave of activity that happens. And then this wave starts at the CA3 and then ends up at the CA1. So there's like a big wave that happens. And this wave is the sharp wave itself. But what happens in CA1 is that the CA1 acknowledges the receipt of this wave. And the receipt of this wave is acknowledged by CA1 creating like short ripples within this wave itself. So imagine there's this wave and riding on top of this wave is CA1 adds its own contribution to this wave and adds like short ripples, small ripples on this wave itself. So imagine, like CA1 adding like a little bit of tiny ripples. So you can imagine it as this large, huge wave that is coming from the ocean. But within that large wave are tiny ripples that are also happening. And this is sort of what's happening as this wave goes from CA3 to CA1. Now, what this scientist Guru do, and again, even I myself pronounce this name different times, different ways, different times. But my apologies if you're listening, dear scientist. But phenomenal study, 2009 study in Nature Neuroscience. And what they found is, first of all, they had a very clever methodology. So what they did is they inserted a stimulation device inside this part of the brain. And what they did is whenever they noticed that this wave is happening, so let's say if the neuronal activity exceeds any particular threshold, so let's say this is the threshold, and if the neuronal activity exceeded this, then what they did is they applied electrical stimulation. So this was like a closed loop process. The moment the activity exceeded the threshold, there was like a stimulation applied to this part of the brain. And what the stimulation does, it just disrupts everything. It disrupts the rhythm of the brain. It adds noise to it. So this process cannot continue. So they did that for one set of animals. And for the other set of animals, they just applied random stimulation. So this is like a very well-designed, well-controlled study. And what they found is quite fascinating, is that when you gate this wave, meaning if this wave reaches a certain threshold, you don't allow this wave to happen. So when you block this wave, when you stop this wave, the process of reconsolidation seems to be hampered. In other words, the reconsolidation does not happen. And the way they measured that is they trained mice in a certain task. I don't actually remember it was mice or rats, but they trained rodents in a certain learning task. So they made the rodents learn something, perhaps a trajectory, to get to a reward. And then on a subsequent day, after the rodents had managed to get some sleep, they applied for half of the population this sort of a disruption in their sharp wave ripples. And for the other half, they just applied random disruptions. And they found that only in the population that had their sharp wave ripples disrupted, that learning performance was significantly reduced. So the next day, the animals could not do the task as well. And this task, if you find this word task very fancy, all it means is they probably trained an animal to walk up to the end of a track and get a reward, something like that. And I think in this task, this was like a bit of a radial maze, meaning there was like the animals had like different arms of this track that the animal would go to. Doesn't really matter the details of the task, but something that the animals were trained to do. And in the next subsequent day, the animals who had their sharp wave ripples disrupted were not able to do these tasks very well. Now, it is important to remember that there was a control group, and this control group also had stimulation applied at random times. But this random time interval group did absolutely fine the next day, and their performance seemed to be improved after this consolidation process. It is also important to note that the following day, the animals that had their sharp wave ripples disrupted, they were not amnesic. They did not forget things. Their performance just did not improve as much, suggesting that this process of consolidation is what was disturbed, not the actual memorization itself. So I think that's quite important if we want to improve our performance. This is probably the first causal evidence, meaning so far everything was just theory and we are observing things side by side, also known as correlation. But when you disrupt this region and you show that what you expect to happen does not happen, this gives us definitive proof that this region was indeed involved in memory consolidation. And if we want our memory consolidation to happen, sharp wave ripples seem to be super important. Cool. So hopefully you understand sharp wave ripples. Let me know again if you have any questions. Let that save itself. I'm gonna causal proof. Okay, actually I like this. So this is also the first evidence that suggests that sharp wave ripples are not. So this evidence also suggests that sleep is not just like a bit of a lifestyle hack, but it might be a necessity if you want to learn and perform better. And later it was also shown that sharp wave ripples happen in rest periods as well, meaning both rest and critically sleep might be absolutely essential if you want your memories to consolidate, if you want your skills to improve, and if you want your brain to perform at the highest level. So sleep is not just a nice hippie lifestyle tip, but there is real science that suggests that it is absolutely critical, not just a nice to have, but might be a must have for good and high performance. Cool, I like that section. Boom. Great. Sharp wave ripples is done. Okay, time has come that we move beyond navigation, because so far everything was about navigation. Now we're going to move beyond it. All right. So far we have been talking about memory in terms of navigation. What is the path that animals or humans take, and how do we integrate that path? But now the question is, does this same mechanism, this same sort of an infrastructure also get involved when we are talking about more abstract things that we code? So for example, we remember very, very interesting things. We remember facts about people, but we also remember other things like social hierarchies, for example. Like you probably know the name of the President of the United States. And the reason for that is there is some sort of a hierarchy that is associated with the position like that. There's some sort of a power that's associated with the things like that. You would probably also remember things like who's the most beautiful person in your high school or something like that. There are all sorts of hierarchies and sequences and paths that we maintain in our brain. And the question is, does this memory infrastructure that encodes navigation, spatial memory, is it the same infrastructure that encodes more abstract things? And the answer to that is a yes. And this was shown in a very elegant, beautiful experiment done by Alexandra Constantinescu. And this was shown in a very beautiful experiment done by Alexandra Constantinescu and others here at Oxford, in fact. And let me show you what they did and how they found some evidence for abstract coding in the brain. So what they did is they showed pictures to human participants. So this is important, by the way. We have already entered the territory of humans. So far we were in the territory of animals. And what they showed is these pictures is like photos of some birds, except this bird differed. So I'm just drawing a bird on the screen. Oops. Do birds have ears? I have no clue. Probably not, but we'll assume it's like a chicken or something. But anyway, they had these birds and there was like a body. So hopefully you can see the drawing of the bird on the screen. And let's give them a tail. So they had these pictures of these birds, but the crucial thing was these birds differed in two levels. So some birds had long necks or shorter necks, and some birds had long legs or shorter legs. So this distance, the neck distance, and this distance varied in birds. So they showed people a bunch of different birds in different orders, and some birds had shorter legs, some birds had longer legs, and the human participants in this task learned that some birds are associated with rewards, and rewards in this case was some Christmas objects. So perhaps this experiment was done in the Christmas time, I'm not sure, but the idea was birds of certain neck and leg lengths got some Christmas objects, perhaps a Christmas hat, for example, or bells. And some birds got a tree, some birds got some bells, some birds got some other gifts, and other birds did not get anything. So what you can imagine is what the experimenters have done, have created this 2D space. And this 2D space is one axis differs in the neck length, and the other axis differs in the leg length of these birds, and some neck lengths and bird leg length combination. and some combinations of neck and leg length yield certain rewards. So some birds get some rewards. So remember, from the participant's perspective, from the human perspective, they have no clue that there is a 2D space being built. What the humans are essentially doing is they're observing that, look, some birds are different than others. Some birds have taller necks, some birds have shorter legs. And this bird with, let's say, the shorter leg and longer length gives me like bells. And the other birds with longer legs and shorter necks give me perhaps a Christmas tree. And some birds in the middle don't give me anything. So the humans are just learning what gives me rewards and what doesn't give me reward. They have no clue that there is like a 2D space that's being created here. And then this is the interesting finding. So you can imagine that this creates this 2D space. Let's say this is leg length, this is neck length, and some points in this space are rewarded. So let's say you start with a bird here with, say, low leg length and low neck length. What the researchers did was asked people to navigate towards the bird that had, let's say, a Christmas bell reward associated with it. So if they started from this position, maybe this bird is what gives the reward associated, and the participants could, increase and decrease the length of the bird so that they sort of moved in this trajectory which takes them to this reward. And what the researchers found, and this is the fascinating thing, is that there's this angle that's created. Let's call this angle theta. And what they found is that the trajectory in which the human participants are moving, so in this case it's this angle theta, the trajectory from one bird to the other that these human participants are moving in also corresponds to the brain signal coming from the entorhinal cortex. So this trajectory, so if you perfectly align with this trajectory, the entorhinal cortex also represents this sort of this angle in which you're moving. And more importantly, that angle is found in repetitions of 60 degrees. Meaning, if you moved along trajectory A, and this is at an angle of, let's say, 10 degrees, then 10 plus 60 would be 70, and a similar signal would be found along the 70 plus 60 would be 130, and a similar signal in the brain would be found at the 130 degree trajectory. Now, why is that thing important? Remember earlier we talked about grid cells, and we talked about how they form a hexagonal pattern. Well, this suggests that if grid cells are responsible for mapping out this coordinate system, then it is exactly 60 degree pattern, the 60 degree repetition pattern that we would expect. And it turns out that even in abstract spaces, this sort of a 60 degree repetition pattern holds, suggesting that grid cells might be the cells that are encoding this abstract coordinate space. So I hope you find this discovery quite fascinating. As much as I do. And this suggests that not only are physical locations in the world encoded by grid cells, so not only are physical coordinates in the world represented by grid cells in the brain, not only do grid cells provide us with a coordinate system for physical space, but it also provides us a coordinate system for more abstract spaces. Abstract spaces like the differences of birds, or perhaps different kinds of people. So you can imagine people differ in various axes. Some people are more friendlier than others. Other people are more popular than others. And indeed, there have been subsequent studies done, including here at Oxford, where they vary different kinds of attributes of people and other hierarchies, and they found like similar patterns of encoding hold. Again, bottom line, suggesting that grid cells might be responsible not just for coding physical space, but also more abstract spaces and abstract hierarchies. Boom. Hopefully that was fun. Hopefully that was fun, everybody. This is so much fun to me. And finally, I would wrap this section And finally, I would talk about this more in the applications section, but this gives us some hints that perhaps a coordinate system might be a memory aid. And indeed, when you see these elite memory athletes, they create like a bit of a mental map first, and then whatever they have to memorize, they put it along those mental maps. And again, we're going to talk about this technique later on, but this gives us some idea that if a coordinate system, a physical coordinate system, is how the brain sort of represents information, then perhaps if we want to remember things, we can indeed use that physical coordinate system that we have already have in our minds, and then attach what we want to remember with points in space that we already have remembered. We're going to talk a bit more about that in the applications. Cool. All right, so now we are ready to talk about emotions. Okay. So emotions. Okay, I'm just going to ensure that you're not hearing a lot of the background noises that I'm hearing. You know what, even if you are, I'm probably be able to remove it and post, so should be. Cool, looks all right. Okay. Okay, recording. We are now going to transition and talk about the relationship between human memories and emotions. So far, we have been talking about what we could call as dry memories. So how do you navigate a path? How do you get to the supermarket? How do you maintain hierarchies in the brain and so on? And while all of that is important, we know that memories have something to do with emotions. We remember big moments in our lives. So for example, recent for me, that was graduation. But we won't remember what happened 2 days before the graduation, and certainly not what I ate for dinner. I might have some vague memories, but even if I do, that's probably an emotional memory. I might have really liked that meal. Similarly, we remember the name of our first crush or the first kiss we have had and things like that. And likewise, on the opposite side, we remember things that are more difficult and traumatic. We remember bad things that have happened. And in the more extreme cases, there are illnesses like either severe trauma or PTSD. You would see it in war veterans who have really faced some difficult situations, life and death situations. And when they return to normal civilian life, it can become harder to forget these memories. So clearly, memory and emotions have something to do with each other. And now the question is, do we know anything about it and what happens in the brain? And does that provide us with any hints of what a therapeutic approach might look like if we were to think about erasing some of these unpleasant, disturbing, and traumatic memories from our lives? So the answer to that, first of all, is a yes. We do know a fair bit about the relationship between emotions and memories. And to understand that, we need to understand two molecules. One is a molecule called adrenaline, which you might have heard about, and the other is a molecule called noradrenaline, which is like a close cousin of adrenaline. Now, depending on whether you follow English or US English and whatnot, these molecules may be referred to as epinephrine and norepinephrine. Same thing, epinephrine is the same as adrenaline, norepinephrine is the same as noradrenaline. Generally speaking, adrenaline is largely found in the bloodstream. That's not like a hard, fast statement, but that's generally where it's found. And noradrenaline is generally found in the nervous system. And in the brain, more specifically, it is also found in places like the locus coeruleus and indeed the amygdala. Now notice that the amygdala is the part of the brain that was also removed in HM's memory surgery. So there's some sort of convergence here, hopefully, that you are seeing. So here is some findings that we have about the relationship with this adrenaline thingy and memory. So first of all, the first finding worth emphasizing, and a lot of this work will be taken from this. A lot of this work will be sourced from this review by James McGaugh, and the specific paper I'm looking at is his review paper in the Annual Reviews of Neuroscience published in 2004. So the first thing to look at is this experiment done by Bohus in 1994, Bohus, and they showed that once you train animal or even people in something, once that training has happened and in the crucial period where consolidation is happening, if you inject adrenaline in their bloodstreams, it seems like the memory consolidation and learning and performance process is improved. So a little bit of stress, let's say, a little bit of activation and a little bit of adrenaline in your blood after training, the little bit of awake state, that state seems to improve performance. Now it's important to note that it's a bit of a U-shaped relationship, inverse U, meaning a little bit is good, but too much might be a bit too bad. But a little bit of adrenaline in the bloodstream after training anything seems to improve performance in subsequent testing. And so that was the Bohus experiment. Again, crucial bit, it needs to be shortly after training. In 1964, though, there was another set of studies, and this experiment I'm referring to is by Goddard in 1964. They showed that if you electrically stimulate the amygdala, now again, this is a part of the brain relatively close to the hippocampus, which pop culture, we think about it as fear center, but as you'll see, it does much more than fear. And indeed, we're talking about the emotional contents of memory in this case. But electrical stimulation of the amygdala in rats, after the rats had been trained on something, they found that this stimulation disrupted this training learning process. And what this suggests is that once you have learned, when you are in this consolidation process, when you are in this building memory process, if you disrupt activity in the amygdala, which is largely associated with emotions, happy faces, neutral, even like and fearful responses, once you disrupt activity in that part of the brain, which is tend to be responsible for a lot of emotional processing, it disturbs the learning and the consolidation and the performance process. So that was an experiment. So Gallagher in 1981 did this clever experiment where he injected adrenaline blockers. So he blocked the adrenaline parts of the brain in the amygdala. And what they found is that once that was infused in rats, it also impaired their training. So it's not just, disruption, but it seems like the adrenaline, the noradrenaline, those kinds of molecules seem to be involved in this emotional process of the memory. So the way I like to think about it is, while a lot of memories are formed in the hippocampus, the amygdala seems to be a little bit of a stamp, an emotional stamp that it provides to those memories, and which determines how important they are perhaps to us and whatnot. So think of the hippocampus as a bit of a librarian, but the amygdala is the one that gives it a little bit of a seal of approval. It's a little bit of a stamp in that it's a negative memory or a positive memory and so on. So another thing that Gallagher did in 1981 was that they infused noradrenaline back into the amygdala. So firstly, they blocked it, and then they infused it back. And what they found was, voila, memory and performance and consolidation, all of these processes were actually improved. And the effects of these tended to be quite significant, even 24 hours after training, suggesting again that the amygdala is involved in this long-term consolidation process. Now, it is important for me to note that noradrenaline is not the only player here. There's also cortisol, there's dopamine, there's acetylcholine. All of these play important roles in determining the emotional valence and the rewarding nature, or the lack thereof, of memories. But regardless, the amygdala seems to be the stamper of emotion on memory. And hopefully this gives you a sense of the circuit that's involved when we talk about memory. And hopefully this gives you a sense of the circuit that is involved when we talk about emotional memories. Now, it is important to think about something called as trauma. Now, trauma, one simple way of thinking about it would be memories associated with fear that we have formed from the past, and we have sort of consolidated from the past. And so you could imagine an instance where the memory gets triggered even when not in that environment, for example, in PTSD, or it gets triggered in that environment. So if there's a particular person who inflicted a lot of harm, and as a result, we have formed a traumatic memory of this person, the next time we do see this person, it's going to trigger that negative memory, and we're going to feel all the fear and the negative conditioning that came from that memory. And this experience can range from a little bit awkward to quite debilitating, causing panic attacks and whatnot. So this is serious stuff, and healing these problems seems to be quite important if we were to live our lives with freedom and more openness and more joy in the world. But also, this sort of a signal is important for us to set boundaries. So now, assuming we want indeed to get rid of these emotions, perhaps we have learned the lesson from it, perhaps we have set appropriate boundaries. Is there any hint coming from neuroscience of how we would get rid of such negative, disturbing, traumatic memories? And indeed, there is a hint. And this hint comes from Joseph Ledoux's lab. And the first author for this paper was Nader, and this is a 2000 paper. And again, this is an absolutely mind-blowing. I invite you to stick with me for this. I'm not going to draw a lot of diagrams, but what I will say is I'll just describe the experiment verbally. What happened is they took rats and they essentially gave these rats fearful memories. So now in the scientific literature, this is called fear conditioning. So what they did was there was like a tone that was playing, and associated with this tone was a little bit of a shock, like a foot shock. Now, I imagine this would not be like super high, and they would have taken care of the animal to some extent. And indeed, the ethics of animal research is a conversation which we should and could have some other time. But for now, let's just follow the experiment. And in this experiment, they played a tone and they give a foot shock to this animal, essentially creating a negative traumatic memory. And then they did something very clever. What they did is later on, they reactivated the tone itself. So what they're doing is making the animal recall this memory. But while there is this recall happening, they actually block the amygdala. So so neurons that fire together, wire together, that kind of thing. What's happening in between neurons in the synapses is there are more receptors that are being built by proteins in the brain. And what they did is block this protein synthesis in the amygdala. And what they found is that if you trigger the memory, but if you disrupt the amygdala during that triggering process, that's when the animal sort of gets rid of that memory. So apparently the animal stops having the reactions to this memory completely. Like they've forgotten this association between this tone and the foot shock completely. And this is mind-blowing because they had completely learned this. This was a consolidated process in other animals where they did not disrupt the amygdala. This was fine. And crucially, this disruption happens when you recall the memory. It's not just any time you disrupt the amygdala, the memory is lost. And so what this suggests is memories are fragile in that they can be changed, and especially the emotional content of the memory can be changed. And the way this can be done, one crucial ingredient of it seems to be, if you think about this study, is you do have to at least bring up the memory temporarily. So if you bring up a consolidated memory temporarily back to the librarian and you disrupt activity in the amygdala, the stamping process, while this memory is recalled, boom, apparently this memory can be extinguished. And as far as I remember, this effect is quite significant. It's not just they sort of forgot something, but it seemed like the fear conditioning was lost. And this study suggests to us, if we were to translate it to humans, and again, there's always risks about doing it this way, but what it suggests to us is that there can be a little bit of a formula to healing. And this is again me saying things rather than this being science. There's more of a theoretical guesswork happening here, which is if you take a memory and if you recall that traumatic memory, but if it is paired with disruption, there is a chance that that process could lead to healing, is what we call it, the word that we use, right? Healing. And again, this is mind-blowing. And if you look at what a lot of psychotherapies do, this is exactly what they do, right? They make you somehow recall and relive those memories, but there is some sort of a disruption process involved. The disruption could be cognitive, like making you really challenge your assumptions. The disruption could be like eye movement. So there's this therapy called eye movement desensitization therapy, reprocessing, EMDR. And what again they do is recall the memory, and then there's a disruption, in this case, eye movements, which tends to reduce the emotional content of that traumatic memory. And this is the kind of therapy that especially works very well with post-traumatic stress disorder. So I'm getting a bit ahead of myself right now because we're going to talk about applications in the next video. But for now, I hope you find this fascinating, which is memories are labile, they're fragile. And if you recall a memory and if you disrupt the stamping process, we have a window of opportunity of erasing at least the emotional content of that memory. And this to me is fascinating. This to me is endlessly keeps me up at night and makes me think about how beautiful this human brain is. So hopefully you learned something today. That was it for today in terms of the mechanisms. What I'm going to do is wrap this stream with a little bit of a recap. Let's do some recap. All right, let's go. So today we covered a lot of ground. Let me get this in frame. So today we covered a lot of ground. We first talked about the idea of the human hippocampus being involved in memory, and we wanted to find out what are the mechanisms that are involved that make memory in the hippocampus possible. So the first discovery that won the Nobel Prize was the discovery of place cells by John O'Keefe. And what they found is that when the rats are freely moving and exploring an environment, there are cells that fire at specific location, at specific places. And these were named as place cells. Later on, Edward Moser and Maybrit Moses discovered grid cells, and they found that the brain maintains a little bit of a coordinate structure. And so you could think of like grid cells as providing the coordinate system for the brain, and place cells as providing specific locations of specific places in the brain. And together this create a little bit of a cognitive map, a map in the brain, which is independent of the animal itself. So remember, no matter where this animal is approaching from, these cells would fire in still the same manner. All right, then we discovered this fascinating phenomenon called replay. Let me get this on screen. Then we discovered this fascinating phenomenon called replay. And what this idea was, if place cells A, B, and C fire, what we noticed that in sleep, when the animal is sleeping, those same cells fire in the same sequence, and they fire in rapid succession. So while this was happening on the order of seconds, this happens on the order of milliseconds. And this is our first hint at what consolidation might look like in the brain. The brain is literally replaying the sequences it has been through during the active phases. And not only was this forward replay observed at rest, at sleep, but there was a reverse replay observed largely when the reward was already collected and at rest moments. So the brain is like playing forward and backward the sequences that we go through. In these cases, these are literal sequences, but we also talk about this might happen for abstract sequences. And forward and behind replay happens, and forward replay also happens at the start, even before the animal has taken steps, but has gone through that path a few times. And this suggests that the animal is already starting to plan its trajectory even before it takes the first step. Again, suggesting like there's this consolidation, memory, learning process happening. And then we talked about something called as phase precession. So while the replay showed us that periods of rest and sleep are critically important, this building block starts to happen in the active exploration phase itself. And you'll see that neurons start firing in places A, B, and C, but the whole sequence also fires, and subsequently if neuron A fired at the top of this sequence, then it goes a little bit behind, almost like making room for the rest of the sequence. Now, something I did not say is it also does forward replay here, but that finding is still in its infancy, let's say, or still not as well established, or the effects are weaker, so I sort of ignored it for now. Then we talked about this idea of sharp wave ripples. And what sharp wave ripples are is there's activity that starts from the CA3 part of the hippocampus, and then it goes to the CA1. But as it ends up in the CA1, there is a bit of a disruption. Well, there's not disruption, but there's a receipt, there's an acknowledgement that the wave has reached me by the CA1, and it adds ripples to it. And within these ripples, first of all, is where the replay happens. And when this activity is disrupted, when the waves themselves are disrupted, the consolidation process stops, meaning the animals are no longer as good as in the task the next day after their training. So what we expect, the benefits of training and sleep, they stop. And this is the first critical evidence that sleep is not just this lifestyle hack, rest is not just this lifestyle hack, but it is an essential component of learning and performance. Finally, we talked about this experiment where they varied the neck length and leg length. And what this experiment effectively showed us is the brain is not just tracking physical coordinates, but it's also tracking abstract spaces. And the same mechanism that is tracking the physical coordinates might also be tracking abstract spaces and abstract hierarchies, like who has more social status, for example, or who's more beautiful, or who's more popular, for example. And then we concluded by talking about the relationship between human memory and emotion. We talked about this critical region called the amygdala, which might act as a bit of a stamp of memories. And we saw that the use of adrenaline, if adrenaline is slightly increased after training, it might improve the memory and learning performance. At the same time, if you disrupt activity in this emotion center of the brain called the amygdala, then you can disrupt a memory formation, especially the emotional contents of it. And finally, we discovered that the emotional content of the memory might be erasable. And this is shown in this beautiful experiment where they take the memory, they form a memory, a negative memory in that. And when this memory is recalled, and at the same time, if you disrupt activity in the amygdala, the animal seems to lose the memory itself. And it seems to lose especially the fear associated with that memory. So hopefully this gives you a good sense of the mechanism of memory in the brain. If you are like me, you would find this fascinating. At least this blows my mind every single day. And it's fascinating to see that human memory is such an intelligent, beautiful, elegant process that happens. And hopefully this tells you a little bit about how it all works. In the next video, we'll talk about the applications. In the next section, we're going to talk about applications. How do we take this information that we know about the brain and apply it to our day-to-day lives so that we can improve our performance, so that we can improve our memories, so that we can remember things that we want to remember, and perhaps even forget things that are not serving us. Again, in the case of trauma, for example. So all of this will be in the next section. Awesome. Let's stop the recording here, and what I want to do now, first of all, Thank you. Thank you. Thanks, Monica. Good to see you here. You are awesome as well. All right, what shall we do today? So what I want to do before wrapping this live stream is just invite you to take the scorecard. It's score.sankalpguru.org. You will find the link in the description. It's in the comment. I invite you to take that so that you will learn about your emotional intelligence and learn about how to improve and whatnot. So that's going to be fascinating discovery. A lot of people say a lot of good things about the scorecard to me. I can't say that it's, like clinically validated and stuff like that, because it's not. But between you and me, I will tell you that a lot of these questions are inspired by clinically validated questionnaires. So a part of my PhD was to study mental health, and I went quite deep into, the different questionnaires. So I think you'll like it. Do take the scorecard. But other than that, we will talk about the consolidation process soon, the application process. And what do we do? What do we do? What is in it for me? We're going to answer that in the next video, hopefully next week. It does take a lot of time to research these things. Hopefully you like it. But other than that, I think it's time to wrap. So thank you very much if you joined me live. Thank you for your presence. Thank you for your interest in science and neuroscience. This is amazing stuff. And as I said earlier, to me, it fascinates me day in and day out what the human brain is capable of, and our remarkable capacity in this case to learn and memorize and absorb knowledge. See you next time. Thank you.